Hello, everyone. I hope you all are doing well. I hope you had a nice weekend. So today we're going to be diving into our next topic of the day, looking a little bit more deeply at genetics, mitosis and meiosis again, and heredity, and how basically things move through, sort of populations as a whole, but also how the human population sort of does its thing as well. In addition to this, we're going to sort of wrap up our sort of final PowerPoint slides on mammalian sort of evolution and animal evolution as a whole, and take a peek at that. And we're also going to talk a little bit about human evolution, how we got to where we are, what are some of the big things that differentiate us from our closest ancestors. Before we begin, I just wanted to highlight a couple things. So we're here today, August 3, which is the date I'm recording this. This is the day the classes. So August 3, I'm going to be recording both DNA and genetics heredity as one lecture, which is what you're listening to right now. We just wanted to sort of make note that your short paper is due the 12th of August, so it's coming up very soon. These classes always come up faster than you think, unfortunately. And then just as a final note, you will have a final exam. It'll be a bit longer than your first one, simply because it's going to encompass 3 weeks of material as opposed to just the first 2. That being said, I just wanted to sort of just take a brief moment to discuss the short paper. And so first, the guidelines here in the syllabus. It's a really very simple paper, basically showing me that you can read science, you can summarize science, write about it, and do it. It's pretty straightforward, and so a couple things: you shouldn't be quoting in this. You should be writing everything in your own words. You should be telling me what exactly this article is. If you want to say something in the article, say it. Don't pull a quote from the article. Just physically tell me what you're trying to tell me. That way I know you're understanding it. The second thing is, please make sure you check with me before you write about your article, because I don't want you to choose the wrong topic, in the sense of I don't want it to be a wrong type of article. So as the sort of assignment says, it's going to be a news article. You can go to a reputable source. NPR is one of the best, as is The Atlantic. But anything like CNN, Washington Post, things like MSNBC, things like that, things that have a reputable thing behind them, are great places to go to get some news articles for science. But the key here is what you're looking for. So let's just do a quick little search on Google, what we can find on climate change. This is a topic that's coming up in a week or so. And so this is, it's pretty heavy on the news. So let's, pop over to Google and we type in climate change, and you'll get a bunch of stuff about climate change, right? You'll get Wikipedia stuff here and so on and so forth. But you'll get information from NASA and the IPCC, which is the Intergovernmental Panel on Climate Change, best panel on climate change there is, the UN, and so on and so forth. But you're noticing these ones are just like, what are climate change? What are the impacts of climate change? What's causing climate change? All those sorts of fun things. Those aren't appropriate. That's not really what you're looking for here. What you want to do is take a dive into the news section, find something that's been picked up by a reputable news source, and see what you can find. So, if we look at just Google News, you can just go to CNN if you chose. So for instance, if you just went to CNN.com and you typed in you guys all know my internet's a little slow from the early classes, but if we typed in, climate change here, we would pull it up and you would see articles about climate change. So you can do it any way you want, but Google News is a pretty good way. And you'll notice if you sort of just scroll down, you'll see news articles picked up by Fizz or The Guardian or NPR. But these aren't really appropriate, these first ones. We're seeing there's New England's, this is about climate change, but it's how climate change has evolved with melting glaciers, COVID and climate change denial being similar, again, not appropriate. How climate science is moving online, again, not appropriate. But if we keep going down, what we're trying to look for is something a little bit more sciencey. And as we sort of get down to the end of the first page, we see farmers across the world are worried about climate change impacting their crops. Now, this is picked up by a newspaper in Colorado, the Denver channel.com. Never heard of it. But one of the things about this is if you sort of scroll down, you read a little bit about it. It's talking about, and you get around their ads. But the key here is, it says Meinhardt's team recently published a study showing 72% of farmers interviewed from around the world were worried about climate change. And this is what you're looking for. You're looking for brand new research done by a group of scientists who started by asking a question, went out and collected data, and answered that question or whatever question they could answer with the data that they collected. This is what you're looking for. There's a news article that was picked up by there was a published paper that was picked up by a news place because it was newsworthy. And this one showing that farmers are indeed worried about climate change, how it's going to impact their crops. You'll see as in a couple days when we talk about climate change, this is sort of a big deal. But this is what we're looking for here. Something that was published, good new science that was picked up by a news outlet. So that's what we're looking for here. We're looking for this type of thing. And again, all those other things I sort of mentioned, not really appropriate. This is the type of thing you're looking for. If you don't find sort of this in your article, a recently published study, it's probably not going to be appropriate. So this is what you're really looking for. Again, brand new science. And the other thing I just wanted to mention is you all had your exam last week. I just want to, I've had a lot of people email me concerned about their exam. I just want to highlight that their, exams are only worth 23% of your grade. And the first exam you took is only 45 points. It was only 45 questions, so 45 out of 115 points. So it was less than half of this 20%. So even if you didn't do that great on this first exam, it's not a huge impact on your first grade. You'll lose a few points out of the 100%, but it's not a huge deal. So I just wanted to put that up. Just so everyone just sort of is, familiar with this idea. But anyways, if you have any questions on anything, please do let me know. With that, we can move on to the topic of the day. And so the topic of the day is going to be first finishing up what we were working on in terms of evolution of animals. And so where we left off, we were talking about the vertebrates. And so these are groups that have spines. And so we spent a lot of time talking about invertebrates because they were the first life on the planet, and they were extremely successful groups of animals. But what we're really interested in finishing up today is this sort of what we consider a higher group of taxa. Taxa have much more advanced features, and they're much more sort of, you could say pretty, but they're much more sort of glamorous, much more evolutionarily sophisticated than, say, a sponge or even something like a sea star that we spoke about previously. And so we sort of ended off by talking about the evolution of jaws and fins, with jaws being a great way to be able to macerate your food, because at this point in time, we only had mandibles. And so you think about mandibles like what an ant has, right, just for cutting. But what we have here is actually physically jaws. And jaws is kind of important, not just as a movie, because that was a pretty important movie if you like movies, but in terms of evolutionary adaptations, jaws are important. They allow you to macerate your food, and as you macerate your food, it's kind of a big deal. You can get more energy out by macerating your food with jaws than just, say, eating it whole and digesting that whole piece. In addition, another really important thing, and this should sort of pique your interest from when we talked about evolution, is the evolution of fins. And so thinking about how the structure of fins is very much set the stage for the evolution of limbs as well. And so fins was a kind of a big deal, allowed you to be more motile, have directed motility, so moving towards one specific direction. And again, we finished off by talking about the things that were struggling moving on to land. As you remember, plants had all sorts of issues when they moved on to land, including gravity as well as drying out. But another issue you face as an animal is simply how do you get air? Because remember, you need to physically breathe if you're going to be an animal, because remember, all animals are heterotrophic, meaning they eat organic material. So remember, you need to sort of solve that problem. And lungs moving away from gills was the way land animals solved it. But in addition, the evolution of bones and modifying limbs to physically hold yourself up from the crushing forces of gravity, as well as preventing your eggs from desiccating out, were all three sort of key things here. But there's a group of organisms that sort of straddles that line, and this is the amphibians. I'm sure you all have heard of the amphibians. We're thinking about frogs, toads, and salamanders. These are terrestrial invertebrates, but they have this sort of half sort of aquatic, half terrestrial life, with terrestrial meaning on land. Now, the interesting thing about our amphibians is they lay their eggs in water. The rest of sort of animals rely physically on a protective shell to protect their eggs, but amphibians have sort of gone away from that. They lay their eggs directly in water, so they don't have to worry about their eggs drying out. Once they lay their eggs in water, they become larvae, and they go through this whole developmental and metamorphic cycle, which then leads them to becoming adults, which ultimately means they're on land. But many of these amphibians, outside of toads, which can sort of live away from water for a pretty extended period of time, pretty much all amphibians are sort of have to live almost in water most of the time because they sort of rely on their skin being constantly wet. So you'll never find an amphibian sort of in the desert. It's always sort of near water, so it's sort of its skin doesn't dry out. So amphibians sort of straddle the line between I live on land, I live in the water as well. In addition, another really successful group of organisms on terms of our sort of evolutionary trajectory was reptiles and birds, which are very closely related. And so we think about reptiles, these are scaly, animals that have scales. And one of the things that's interesting about our reptiles is that, and you all probably have heard this before, but they are cold-blooded, meaning they are exothermic, meaning that they totally rely on the environment around them to keep them warm. So if you are a chameleon, like we have in this picture here, this chameleon solely relies on the heat of the sun and the environment around it keeping from essentially dying, keeping its body going. And the reptiles and lizards are a pretty diverse group of organisms. As you can see down here, there's about 8,500 or so reptiles on the planet as well. That's in contrast to birds, which are the closest relatives to reptiles. Birds also were the closest relatives to dinosaurs. Reptiles are not the closest relative to dinosaurs, it's actually the birds, but I'm sure many of you have probably heard that by now. Birds evolved the capacity for feathers, so it's just sort of a precursor to hair. And this, sort of the interesting thing about our birds is they are the first sort of vertebrates to evolve flight. So they took this body plan that, again, would have descended from fins, because remember, we're coming from the water to the land. This would have descended from fins, and they took that design of the fins and adapted it for flight. And flight, in terms of these birds, you're thinking about very light, very sort of hollow bones that allow them to be lighter than you would imagine, which allows them to take flight. Now, the idea that's a key difference here between reptiles and birds, and what really separates a lot of animals from reptiles or even invertebrates, is this capacity of being what's called endothermic, meaning you generate your own heat. So as you're doing your cellular respiration, you produce extra energy. And this extra energy is in the form of heat. And this extra heat keeps birds warm, keeps us warm, keeps anything that we would consider to be warm-blooded warm. And this is sort of one of the key components about being a mammal. And so mammals, as birds are, they're endothermic, meaning they make their own heat. And they actually, and then that, again, that's in contrast to being exothermic, which, like reptiles and amphibians, completely relying on the environment to keep you warm. And now when we're thinking about sort of evolutionary features, again, birds, they evolve these feathers, and feathers they think are one of the precursors to fur, but it's sort of a hard thing to sort of infer evolutionary. But when we're thinking about sort of early mammals, thinking about things like, were around by the time of the dinosaurs, you're thinking about things that were longer legs, very low to the ground, very small evolution of hair. And then the important thing about hair is the capacity to trap all this endothermic heat that you're generating relative to, say, any other thing. So early mammals, small, hairy, low to the ground, again, very, very small overall. And overall, our mammals are divided into sort of three major groups. We have our monotremes, and our monotremes are a very, very, very small group. There's only 5 species of monotremes on the planet. This includes the platypus, or you might have heard it as the duckbill platypus. And that's one of the species that's found in Australia. Really, really weird species. They're venomous, the only venomous mammal out there. Again, really weird species. They have spines. And then there's also 4 species of what are called echidnas, and they are, again, also very weird. But these animals are the only mammals that actually lay eggs. And what's sort of interesting about these groups of organisms is they actually produce milk, but they don't actually have nipples. They have these, essentially these hairs that the milk sort of secretes out onto, and the babies just sort of lap it up. Next up is our marsupials, and this is kangaroos and that other hopping group of things that is escaping my mind right now. And sort of interesting thing about our marsupials is they do give birth, but their sort of gestation period inside the womb is very, very short. And so the little baby kangaroos, joeys, as they're they're called. They come out and they're very, very tiny. They're almost partially undeveloped. And so the mothers and the marsupials essentially stick them inside these little sacs they have and carry them around, and they essentially develop outside of the womb while essentially feeding them with milk. And then finally, as in terms of our mammals, we have our placental mammals, and this is where we reside. The picture of the chimpanzee should sort of hint at that, but these are very different. As you all know, we produce, and monkeys and any other sort of primate or any placental mammal. Again, there's about 4,500 species of them, but the idea being that this placenta is a very rich and nourishing thing, provides oxygen and nutrients to the embryo as it develops from single cell all the way up to, again, hundreds of billions of cells. And again, it's all done sort of inside the human body, which is in contrast to, again, eggs, or as developing a sac like a marsupial. So those are the sort of major evolutionary milestones along the way. We really started talking back here about 500 million years ago, and we sort of just expanded, thinking about some major things that we talked about, including the evolution of a spine, jaws, lungs, legs, the capacity to live on land, amniotic fluid, hair, feathers, things like that. So there's lots of things that have evolved along the way, and as you'll notice, that again, that tree of life is pretty diverse. But in terms of the planet, it's only been around for about 1/8 of the planet's history. So it's evolution that's happened really, really fast. And one of the things that you should sort of just take note of is that, remember, this has really happened after oxygen became a thing, right? After the great oxygenation event, evolution really took a foothold, because oxygen is this great sort of life giver. It provides all sorts of brand new and funky paths for things to go down. So that is our sort of wrap up about animal evolution. So let's transition now to talk about the genetic code and genetics. But sort of related to what we just talked about, we're going to talk about how humans evolved, how primates evolved. And so, as I'm sure many of you heard, we all belong to the same genus species, Homo sapiens. There is no subspecies of human. There is no strain of human. We're all just Homo sapiens. And so we all belong to one genus species. If you remember back from our sort of talk about what defines a species, again, it's the capacity to reproduce and produce viable offspring. So that sort of keeps us there. We have the capacity to make no viable offspring with any other species out there. The idea with humans, we are essentially what are called the last extant hominid species. And when you hear extant, that's in contrast to extinct. So extinct meaning completely wiped off the planet, while extant means living. So we are the only living, breathing hominid species, and hominid being human-like. And so you could think of, maybe if you've watched the History Channel at night, and they start to talk about cryptozoology, right, talking about Bigfoot or Sasquatch or something like that, that would be considered a hominid as well. But as far as we know, there are no other extant hominid or human-like species out there, just us. Now, our tribe, which is called Hominini, which includes us and chimpanzees, is something that likely came to be about 8.5 million years ago. And then we diverged from chimpanzees between 4 and 7 million years ago. Sort of a difficult thing to pin down. It's just, we're looking at fossils, we're looking at DNA to try and interpret these things. So it's in the ballpark of 4.5 to 7 million years ago when we split from chimpanzees. And now we can look at sort of our relatives. And so at some point in time, as we can imagine, there was some sort of ancestral primate, and natural selection did its thing, right? These things started living in different habitats. So maybe you had a tarsier, which are really creepy looking in my opinion, but they started living in trees, and you had monkeys living in trees, but then you had larger animals like gorillas or even chimpanzees being mostly on the ground, right? So the idea being is these species diverged evolutionary speaking because they were living in different habitats, right? And living in different habitats allows natural selection to select for different traits that are beneficial to your habitat. Now you can think of something as very different as a human compared to say a New World monkey here, right? You can think of something, well, okay, that's pretty different, right? This monkey lives in a tree, it has a tail, right? It has very different features, but humans were bipedal. Original humans, we think, were in grasslands and stuff like that. So there's a lot of sort of major differences between these groups, but we do all share the same ancestor. We all have a very similar set of characteristics that all point, again, to that common ancestor. And in terms of evolutionary time, it hasn't been very long. You can see that our ancestral primate was somewhere between 55 and 60 million years ago. So it's not a very long time since we diverged overall. But in terms of thinking about hominids, humans, as I mentioned, are the only current extant species or living species of hominids. But that just means for us to exist now, there was something that happened before us. And in fact, if you look at all the sort of related fossil evidence that we have, there's a pretty rich tree of hominid evolution that sort of predated us, that sort of led, you can see evolutionary adaptation sort of accumulating over time. And so you can see, us sitting here in modern times, as well as chimpanzees being our closest relatives. But you can see some other species that you may have heard of, maybe like Neanderthals, Homo neanderthalensis. Only went extinct maybe 15 to 20,000 years ago. Or maybe you've heard of Homo erectus or Homo habilis, these species that went extinct much more distantly in the past, but again, still very closely related to us. But what you'll notice is that we are the only living members. So we know these existed from the fossil records. And we know we are very closely related to chimpanzees by both shared characteristics as well as DNA evidence. But you can notice that, again, lots of things had to go extinct for us. And one of the sort of interesting things is actually when you start to look at the genomes of Homo sapiens, you actually start to notice that a fairly decent proportion of the population, in particular those that live in Europe, have a lot of Neanderthal DNA actually physically in their genomes still. So there clearly seems to have been some cross-mating between these groups. But at the end of the day, we're the only people that really made it out the back end. Everything else, again, exists in the form of fossils. So we can think about what's the evidence? How do we know humans have this rich fossil record? How do we know we evolved from chimpanzees? How do we know we share relative with other apes? There's lots of evidence here. There's fossil evidence. We can look at our DNA, and chimpanzees, as I mentioned several times, we share 95 to 99% of our DNA with chimpanzees. So we're very, very close. In addition, we also share a lot of behavior with our chimpanzees, capacity to think, capacity to use tools, bipedalism, things like that. Lots and lots of evidence out there for sharing common ancestors with chimpanzees and being very, very closely related to chimpanzees. This website, I forget the name of the university, has a really nice sort of X section here about fossil evidence and all the sorts of evidence we have about humans evolving from apes. So if you're sort of interested more in this, this is a really great resource for you to take advantage of. But it's sort of beyond the sort of scope of this class for me to dive really deep into this sort of evidence here. But we can look at lots of sort of differences, again, looking at differences in traits here, and human and apes share a lot of things in common, bone structures and the way we walk and the way we behave and things like that. But there is lots of distinct things that differ between us, right? And you can look at something very, very simple as say looking at skull structure here, and you can look at how vastly different humans are from our closest chimpanzee relatives. But you can also see how much more developed our brains are, and this is what the sort of the major evolutionary thing about humans is how large our brains are relative to our body size. Now, other things might have larger brains simply because they're huge, but relative to our body size, we have huge brains. And this huge brain allows us to do all the things that we do, right? You're able to listen to me in class because you have this brain. If you had this little dinky brain here, you wouldn't be able to do very much. You wouldn't be able to understand what I'm doing. But this brain gives us all these new behaviors. It gives us all these new capacities to do tools and things like that. And that's really the main sort of key feature here. But there's all sorts of other things here that we can see that's really different from between apes, whether it's, body shape, the brain size, length of our limbs, things like that. So there's all sorts of differences, but the key being here is this brain. This brain allows us to be humans, right? There's lots of smart animals on the planet, whether it's a raven or a chimpanzee or a gorilla, but the brain sort of separates us really distinctly from all these as being just that much more advanced than everything else. So back to this sort of thing, we can look at sort of this timeline. We can look at, okay, we shared an ancestor chimpanzees about 5 million years ago. Again, depends on who you ask. And there's lots of things that went extinct. But what is sort of interesting is we can look at sort of where we find our fossils. And so again, this is going back in time. And you'll notice that many of these species were around for, short or long periods of time, depending on essentially, what we could find for fossils. I mean, this one might appear that it was around for longer, but it might just be that we couldn't find fossils here or here. So fossils as well as outside of Africa. And so the idea being here is that where did we come from? And there's sort of two leading hypotheses here. The first is what we call the multi-regional hypothesis, and the second is what we call the out of Africa hypothesis. Now, they're very, very similar, but the key difference here is that there is something, the multi-regional hypothesis, the idea is that being Homo erectus, so if we go back here, Homo erectus being the most closely related ancestor to us and Homo sapiens, evolved in Africa and then left it and sort of dispersed pretty widely. So that's the first one. That's the multi-regional hypothesis that, you know, Homo sapiens evolved in multiple different places, but they all had that one common ancestor that left Africa, and that organism that left is again was Homo erectus, and that's the multi-regional hypothesis. The second one is the out of Africa hypothesis, and this one has been almost, I mean, it's not settled science, but it's more or less true to the vast majority of people that work in this field. But the idea being is that instead of Homo erectus leaving Africa and Homo sapiens evolving multiple times across the planet, Homo sapiens first evolved in Africa and then spread out of Africa. So Homo sapiens, as you can see here, evolved about 100,000 years ago and then left Africa, which is why the idea being is that, you know, maybe if this happened, it wouldn't be as, you know, humans are so similar, even, you know, those that live in Australia and Asia and in North America, is that if the idea being is if this potentially happened, humans would be people speculate that humans would be much, much more diverse. But the fact that we're all so similar really tells us that it's most likely Homo sapiens first evolved in Africa and then spread out. And the way we know this is actually by looking at the mighty mitochondria, so the powerhouse of our cells. And this is sort of a nice summary article about sort of this that came out a few years ago. But this is how we know pretty strongly that this out of Africa hypothesis is most likely true. And so if you remember the mitochondria, it's the powerhouse of the cell. It has its own DNA. And one of the things that's interesting about the mitochondria, as we've mentioned, is it replicates its own DNA by itself, and it replicates itself during mitosis as well as meiosis. And so if you remember back to sort of a few classes ago when we talked about sperm and egg were made, sperm are very simple, right? They only have a capsule, or they have essentially a membrane around them, and they have a tail, and they have the DNA. And that's pretty much all sperm have. Sperm have no meaningful amounts of organelles or things like that. But if you remember, our eggs that we make are huge, right? They're massive, right? They're hundreds of times bigger. They're about 100 times bigger than the sperm. Now, as I mentioned, the egg essentially creates all the sort of starting vesicles for life, including these organelles, right? And the idea being is that you get because it's making all these organelles, the egg also creates the mitochondria. And remember, once you get that fertilized egg, mitosis takes over, right? You're making copies of that same original cell over and over and over again. That being said, the mitochondria you make is a direct descendant from your very first cell that was ever you. And remember, you got that very first cell by essentially being mostly comprised of your mother's cell. So your mitochondria is directly descendant from your mother. And so we can use mitochondria and the DNA inside them to trace lineages back, because we know that these are passed on from mother to children. And it doesn't matter if you're a girl or a boy, you get your mitochondria from your mother. So we can use mitochondrial DNA to trace human lineages back. And when we do this, this is from a really nice paper. We can see this is just a really sort of funky phylogenetic tree. It goes like this, it bends around the corner, and it's like this. And these are what are called sort of distinct subgroups or distinct haplotypes. It doesn't really matter what the sort of jargon here is, but what you'll notice is that at the base of the tree here is red, which is all Africa. And what that really tells you is that, well, if the mitochondria of Africa sort of forms this one group, and we all descended from, again, one sort of single group of mitochondria, the idea being is that whatever forms the base of this tree Africa in this case is the most ancestral and thus evolves first. So the idea being is that we can use the mitochondria to infer that human life essentially evolved in Africa first and then spread out. So that's the idea. Life, by using the mitochondria, we can say humans likely arose in Africa and radiated out. What you'll notice is there's all sorts of different groups, whether in North or South America or Asia or Europe. But so evolution has taken its sort of foothold in these different regions, which gives us all our sort of different characteristics and traits. But again, we all sort of originated from this ancestral population here in Africa. Again, we know that all because of our lovely mitochondria. And once we sort of evolved, we spread relatively fast across the planet. We 200,000 years ago, we were in Africa. We spread to Europe within 100,000 years. We made it all the way down to the Pacific Isles, several thousand years later, and eventually over into North America much more recently. So we spread pretty quickly, but again, we first started off here in Africa. And one of the sort of interesting things about humans, if we look at our population size in about 100,000 BC, there was only about 10,000 of us. And since about 100,000 BC, we know there's been approximately 3,000 generations. And if we look at that sort of the modern day population, there's about 6 to 7 billion of us. Well, actually, there's 7.54 billion of us on the planet. And so one of the things that's actually interesting about the human population is we started off pretty small. Very, very small numbers. We know that there's about 100,000 there's about 10,000 and 100,000 BC. It's estimated that the very original starting human population was only about 10,000 individuals. And we grew really fast. And we grew in from 10,000, or you could say 100,000 10,000 about 100,000 BC. And within 100,000 years, we have billions of us. So we grew really fast for a very large species. And one of the sort of interesting things about humans is our genomes actually have very low variability. So relative to other organisms on the planet, there's very low genetic diversity in humans. And it's not because we don't evolve the same way other organisms do, but the key here is that we started small, and small populations lead to smaller amounts of genetic diversity. And so one of the things is, again, humans are evolving. I don't want you to sort of take that away, that humans have stopped evolving from sort of our ancestral populations. We definitely are. We just started with a very, very small population size relative to sort of other groups of organisms, which leads to our low genome diversity. That being said, we are evolving, according to most people, we're actually evolving faster now than we ever have in the past. So I think that's kind of a fun fact. So we can ask the question next is why the change in genetics between populations? Well, these should be pretty essentially common sense to you at this point. Thinking about evolution for a couple weeks now, adaptations to new environments, food production led to new diets, technological advances, as well as population increases, which brought along with it disease. As we're seeing with COVID, disease is a major check on society. So all these things allowed changes to happen in populations, which leads to the sort of the very distinct characteristics we have between regions, right? People that live in Europe look very different than those that look in Africa, or in China, or in Japan, or in Australia, right? We all have these regional characteristics that all arise from natural selection, right? Changes in the environment, selecting for traits that are beneficial. And as we'll see later in the week, not only just traits, but also behaviors. So I think that's sort of an interesting thing about, because we all have very different behaviors between different cultures as well. And so with that, I was going to talk about the Human Genome Project, but it's not all that point. We know so much about our genomes, about our evolution by sequencing our genomes, and we know this was done in the early 2000s. It was a pretty exciting period in American science. One of our last sort of great hurrahs that we've done as a country. 2001, we know that we sequenced the human genome, and this sort of led us to sort of understanding all these things that we just talked about. And just as a reminder, remember how you get new traits into populations, how populations diverged, how the human population diverged. Again, it was all through mutations, changes in their DNA. So with that, that's going to be sort of our last bit about human evolution. If you have any questions about it, I just want to give you sort of a shallow dive, but if you have any questions, please let me know. So let's go back to the central dogma of biology. Let's get to thinking about our sort of central dogma, right? We have DNA, which is the mechanism by which all life stores information. We have RNA, which is that message, and then protein is ultimately the action. But So that's the central dogma. Keep that in your mind. And remember that your DNA is stored at least in your cells, it's stored in the nucleus. And the nucleus, it's just, remember, it's a membrane-bound organelle that houses most of the DNA in your cell, with the rest of your DNA in your cells being found in your mitochondria. Now, remember your nucleus here houses your genome. And remember, your genome is comprised of small sections of DNA called chromosomes. And humans have 23 pairs of chromosomes, so 46 in total. And remember, on those chromosomes, we have genes, which are essentially the instructions to make specific types of proteins, which again are the action within your cell. And just as a reminder, again, 23 pairs of chromosomes, and every chromosome is paired. So if you have chromosome number 1, you have 2 chromosome number 1s. So you essentially have 2 identical or homologous pairs of chromosomes. And remember, when you have homologous pairs of chromosomes, it means they have the same set of genes on each chromosome. So every time you have a different type of gene or the same gene on 2 different chromosomes, those are what we call alleles. So you can have, essentially, allele can be identical between 2 chromosomes, slightly different, or very different. And in rare cases, they could be absent. You could have just one allele and not have another allele on another chromosome. But just as a reminder, you have 2 types of cell in your body, right? You have your non-reproductive or your somatic cells, which have a diploid number of chromosomes. So they have 2n. So in the case of humans, n is 23, 23 being the number of chromosomes, or n. When you are diploid, you are 2n, meaning you have essentially 2 times the number of chromosomes, or 46. Now that's all your somatic cells. That includes, you can see it's part of your immune system, it's your neurons, your skin, your liver, your bladder, so on and so forth. Now that's in contrast to your gametes and spores, which are sexual cells. In our case, they're just gametes. Spores are a plant and fungal thing. But their haploid number is n, or 20. In the case of us, it's 23. So again, 2 very different types of cells in your body, and remember, they are linked together. And so remember, when you are, when your parents made you as a kid, they were diploid individuals. They had 23 pairs of chromosomes. We had a biological male and a biological female come together. The biological male producing sperm and the biological female producing that egg. And remember, these are gametes, meaning they're haploid, meaning they have half of each. They have half the number of chromosomes. So, they have 1 chromosome 1, 1 chromosome 2, and so on and so forth. Now, the idea being, if fertilization happens, sperm meets egg. You guys have all heard of the birds and the bees at this point. And when that happens, fertilization produces a zygote, which is a fertilized egg. And this fertilized egg now is diploid. And so you'll see now it has 23 pairs again, and it's 46 chromosomes in total. And I think it's pretty obvious to sort of point out that half those chromosomes came from your biological dad, and half those chromosomes came from your biological mom. So this is what we're doing. And then once you have a fertilized egg, mitosis takes over. And mitosis, again, is just replication of cells. So making a copy of your genome, making a copy of the organelles, dividing, 1 cell becoming 2, 2 becoming 4, and so on and so forth, till you have enough cells to make a living individual. So that's the flow we're thinking about. So keep thinking about that central dogma. Keep thinking about this flow of information. But let's talk a little bit about meiosis. And let's talk about an important part of meiosis that actually helps with genetics. And so we talked about interphase, and interphase here on the left is our part where the cell replicates its chromosomes and its organelles, gets ready for division, and it's growing. Now, remember, meiosis has 2 steps, meiosis 1 and then eventually meiosis 2. Now, meiosis 1 has a very, very important process in it. There's a step in meiosis called prophase 1. And in prophase 1, what ends up happening, we have this process called crossing over. And we're going to talk about this in a moment, but let's just keep this in mind. It's very, very early on in the meiosis process. And you'll see that this crossing over essentially is swapping of pieces of your chromosomes. So essentially, if you sort of use this cartoon depiction, you see there's dark blue here and light blue here. They just had essentially a swapping of chromosomes here. And you see the mitosis process unfold, like we talked about, producing 2 identical daughter cells, and then those daughter cells do not replicate their genome again, and they divide again, leading to that haploid number of cells here. But let's just sort of put this in perspective. So we'll take this handsome-looking pit bull here, and we'll just sort of put this in perspective for a human life cycle, which again would be very similar to a dog. Right? We have these haploid cells, sperm and egg. and they are fertilized to form essentially a fertilized egg, which is a zygote. And then mitosis takes over, creating this, again, this very handsome pipple here. Now, this pipple and its partner eventually would undergo meiosis to produce their gametes, which would cut the amount of hereditary material in half, producing these haploid gametes, allowing the cycle to repeat over and over again. So this is the flow we're thinking about, right? Mitosis takes over after meiosis, produces essentially the gametes here, and once the gametes are produced, one can fertilize the other. So this is the flow of information we're thinking about. And just as a reminder, this is what your DNA looks like. You have 23 pairs of chromosomes. This is what we call a karyotype. This is a male. So you see we have those 22 regular pairs of chromosomes. But in addition, we actually have a pair of chromosomes down here called your sex chromosomes. So the X and the Y that I'm sure you all have heard about. Now, these X and the Y are, your sexual chromosomes. The remaining 22 pairs are what we call autosomes. They basically just mean they're involved in no way with determining sex. All biological sex is actually just determined here on the Y chromosome. But let's get back to this idea of crossing over. And so again, the idea during meiosis 1, the very first step, we have 2 sister chromosomes. And during this process, they cross over, again, swapping bits and pieces of each other with one another. And eventually, if you think about sort of how these chromosomes are, you're swapping potential alleles, right? So you can imagine if you have one, let me actually get to a better example here. So you can imagine if we have 2 chromosomes here, and we have a couple of different genes here, A, B, and C, right? We have a crossing over event. Well, maybe by crossing over, we have, instead of having 3 capitals and then 3 capitals on the other side, well, now we have capital and 2 lowercase. And the same thing over here, right? We had 3 lowercase, now we have 1 lowercase and 2 capitals. So the idea being is you could potentially swap alleles between these chromosomes. And what this really does is, if we sort of go back to this diagram, is that these alleles end up getting, actually, let me go back to this diagram. What you'll notice is that these alleles would ultimately get sorted into one or the more daughter cells during meiosis 2. So what it means that is, by crossing over, you're adding essentially novel genetic diversity by essentially swapping the order of things, potentially putting types of alleles together that never were. Again, the very simple case here is the capital and lowercase things, but putting things that are different together that weren't there before. And then that's an important thing, because by essentially shuffling, you think about, you take a deck of cards and you shuffle them, you get this brand new potential order. That's the same thing that's going on here. We're shuffling our genes a bit and getting a brand new potential order. And what this does is it adds all sorts of new and novel genetic diversity to your gene pool. And this is good and bad, right? Genetic diversity is not always good, right? It can be a bad thing if you bring together bad things, but it is generally considered a very good thing. And what it actually does is it makes you as an individual 1 in a trillion. And so what this does is this crossing over event in sort of all on its own creates, every time an organism produces gametes, each one is 1 in a trillion. It's almost entirely unique, which I think is a really sort of interesting thing, because it makes you 1 in a trillion, right? It makes you, as you are, that order of genes, very, very unique. It also means, because it's essentially, very unique, the idea being is that you can be very different from your siblings. So if I had a picture of my sister and I here, you probably would never have guessed my sister and I were essentially from the same parents, because we look very, very, very different. But again, this is all due, not to the milkman or anything like that, but due to this idea of crossing over. Introduces brand new novel traits by just shuffling the deck of cards that is your genome. So it's a really important process during meiosis, again, that adds all sorts of nice novel genetic diversity. In addition, it also adds lots of genetic variability. So we can look at a litter of cats here, and we have the mother cat here and her 4 offspring. And again, the mother cat would have been impregnated by by some male cat. And what you notice is the 4 cats that come out the back end are crazy, crazy different, right? We have the orange tabby and the gray tabby and black and white, right? This is all due to crossing over, sorting genes, sorting alleles, creating new trait combinations, and that's what we're seeing here. You see that with cats, you see that with humans, you see that with anything that does meiosis. So meiosis is a really powerful way to introduce new genetic diversity to a population. But meiosis is not perfect. And so during meiosis, we can have a process called non-disjunction. This is simply the process of meiosis failing to separate chromosomes. So as we mentioned, during this process, the chromosomes are split. So half goes to one side, half goes to the other. Now, sometimes homologous chromosomes don't separate, as we see here in this cartoon example, where we have essentially an organism that's missing half the amount of DNA that it should, or that has essentially 50% more DNA than it should. Most commonly, errors in meiosis just rely on essentially one extra chromosome being added to the pool. So you have, in this case, you have one extra chromosome in each of these gametes, where these chromosomes are, this gametes here are essentially deficient. And that's pretty common. It depends on how old you are as an individual. So older folks are more likely to have non-disjunction errors like this. But one of the most common cases of non-disjunction we see in sort of our current society is Down syndrome. And so this is trisomy of the 21st chromosome, which is down. You can see on this sort of smearing of chromosomes. We have 3 21st chromosomes, and this essentially was a form of non-disjunction. This 21st chromosome failed to separate, and this person ended up with 3. And as you can see, having one extra chromosome can have some pretty dramatic effects on the biology and the behavior of an individual. Those with Down syndrome have all sorts of varying biology about them, in addition to sort of their overall change in behavior. So non-disjunction in this errors of mitosis can lead to some pretty serious issues. In addition, as I mentioned, your chromosomes tell you about your sex. Now, when we're thinking about sex, we're thinking about biological sex as you are born. And there is an important distinction here, biologically speaking, in terms of science, to differentiate between gender and sex. There is a pretty stark definition between the two. Sex is something you're born with. Gender is more of an identity. It's more of something that is mental in capacity. It's not something that's really hardwired into your genetics. And there's a lot of sort of psychological research about that if you're interested in any of that. But just to know, when we're talking about anything with sex in this class, we're really thinking about just biological sex. Gender and that sort of aspect, it's not as sort of concrete as, as biological sex is. So just to know, that's what we're talking about here. And if you want to, if you're interested in any of this stuff about how, the psychology of gender and stuff like that, please let me know. There's all sorts of research out there about it. I'd be happy to share it with you. But again, for the purposes of this class, we're talking strictly about biological sex. In humans, as with most animals, it's determined by your sex chromosomes. And the idea being is these chromosomes have genes that are just strictly involved with determining your sex. That's pretty much it. They don't really do too much else. The Y chromosome does. The X chromosome in males and females do that, but the Y chromosome really is only involved in making males. And sort of one of the interesting things about the Y chromosome, it is very, very small, as you can see, relative to the X chromosome. And one of the sort of interesting things about the Y chromosome is that it's really not just having the Y chromosome, it's just the right genes on the Y chromosome being activated that make you a biological male versus a biological female. So, further interesting thing, the Y chromosome, very tiny but powerful in determining sexual sort of identity. And sort of one of the things you can sort of think about is that every human being was a female at some point during development. And eventually the genes on the Y chromosome took over and changed a male into a female. So it just made us grow genitalia, different types of genitalia. But that's why we have nipples. That's why you're essentially developmentally the same. But again, it's some certain point that the Y chromosome really takes over. Sex determination in humans, again, is pretty simple. Y chromosome typically makes male because of the genes present. But there are some sort of oddball types of biological genders out there. And so these are thinking about, we can have all sorts of combinations. You can have, XXY, XY, and so on and so forth, right? You can just mix and match combinations of chromosomes with one another. So sometimes we can have individuals that have just one X chromosome and no Y chromosome or no second X chromosome. And these individuals typically develop what's called Turner syndrome, leading them to shorter heights. They have these really sort of odd looking webs of skin develop between their neck and their shoulders, and all sorts of problems with their sort of ovaries and sterility, and as well as potential some learning difficulties. Those that are XXY typically develop Klinefelter syndrome, which leads you to have underdeveloped testes, lower testosterone. Typically, you're slightly more feminine, and you typically have longer limbs, and you're slightly taller than average. So think of someone that's kind of like, a little bit ganglier. We can also have super males, which are XYY. So these are males that are typically taller than average. They typically have very high levels of acne, and they're typically slightly less smart than your average everyday human, but otherwise they develop normally. And then finally, we have what are called metafemales, which are triple X, and they may potentially be sterile, but they're not always, and there doesn't seem to be anything sort of different. So you can have all sorts of arrangements of your sex chromosomes, but in terms of strictly definitions of biological male versus biological female, it's really the genes on that Y chromosome that differentiate them. So in terms of other animals, there is all sorts of other ways out there that you can become a male and a female in other species. So we'll talk about the bird system. They have the WZ system, and it's sort of interesting. In birds, the mom determines the moms as opposed to the dads determine sex. So you'll notice here that it's, females are WZ and males are ZZ. Anytime you have the W chromosome in birds, you're a female. If you have two Zs, you're a male. And so the W chromosome is essentially akin to the male Y chromosome in humans. Next up is bees and wasps. And most of this group, bees, wasps, ants, these are called the Hymenoptera. These are our arthropods, again, very successful organisms. But you notice is that our females here have a full set of chromosomes, while as the males only have half the number of chromosomes. But it's sort of interesting thing about these societies of, ants, bees, and wasps is that the males are only around for a very short period of time. And they're really only around for sex. That's really all males in this group of organisms do. And so it sort of kind of makes biological sense that females will have the full set and males, which again are only really involved in sex, have half the set. And then finally we have the turtles, and the turtles are reliant not on chromosomes, but on temperature. So hot eggs turn out to be female, cold eggs turn out to be male. And there are some other systems in insects and chickens and stuff like that. So there are other types of systems out there, but these are the most common ones overall. And so that's meiosis. That's sort of thinking about heredity. But let's talk about mitosis. So when we think about how you went from a fertilized egg to the adult you are now, it's all mitosis, right? Taking one cell, making it two to four, 4 to 6, and so on and so forth, right? That's how we got from single cell to multi-trillion cell. Mitosis is also how we repair cells in our body. And so we can ask the question is, what happens if mitosis goes crazy? So we talked about issues in meiosis. What happens if mitosis goes crazy? And this one probably sort of touches the hearts of everyone in this class, but when mitosis goes crazy, not so fun things happen. So mitosis as a process, so sort of this multi-step process, is heavily, heavily, heavily regulated. Basically, like there's a bajillion and one checks along the way that if your cell fails any one of them, it essentially stops mitosis, prevents the new cell from being created. I apologize for that. I had a puppy issue I had to take care of, but as I was mentioning, mitosis is a heavily regulated process. Any sort of step along the way here will essentially lead to mitosis being terminated. Now we can ask the question is, one of the ways that cells, before I get ahead of myself, one of the ways that cells actually will stop mitosis from going crazy is they'll actually kill themselves. And so this is a process called apoptosis, which is programmed cell death. Your cells will actually kill themselves if they're not performing mitosis correctly. We can ask ourselves now is what happens if cells have unregulated mitosis, and this is what we call cancer. So cells that divide uncontrollably, do not care if mitosis is making errors or things like that, are called cancerous. And these cancer cells essentially ignore all checks and balances while they're performing mitosis, and they also do not listen to cells around them. And sort of the last thing I'd like to mention about cancer cells is they do just arise from genetic mutations. Typically the genetic mutations that occurred in the sort of the genes or the functions that are associated with mitosis. So there are other genes that are hit, but these are the most common, the mutations in genes that regulate mitosis. The idea being is if you have normal cells versus cancer cells are oddly shaped relative to normal cells, just as a note. Normal cells will divide and do their thing. and they'll be checked. They won't divide uncontrollably. If they make errors during mitosis, they'll kill themselves, and they'll eventually stop. Cancerous cells, they don't care. They make errors during mitosis. They will keep dividing indefinitely, basically, and incidence in the population. So some cancers like prostate and breast cancer or even lung cancer are very, very common in the population, but other types of cancers, potentially like pancreatic cancer or, sort of weird cancers that you've never heard of, are much less common, but can be deadly. And so it doesn't, certain cancers that are pretty common, like prostate and breast cancer, are less deadly simply because, things that are really, really abundant in human populations are much more heavily studied. So we have much lower death rates here. But some things that are even really abundant are very, very deadly, like lung cancer as well. And as you guys probably all know, once you have a tumor form, and if that tumor ever spreads to any other part of your body, your tumor has undergone metastasis, basically just spreading. And so you can look at this, a screen imaging of this person here that has a tumor here, but that tumor here is spread all throughout their body. And again, it's hopping on that circulatory system. Sort of the unfortunate thing about metastasis is that it doesn't really matter what cancer you have. If you have prostate cancer and you don't know it, and it metastasizes, it's most likely means that you're likely going to die from it. It's not very common people come back from metastatic cancer, unfortunately. But we're thinking at a biological level what this looks like. We look at, we have a blood vessel here, we have normal cells, and we have cancerous tumor cells here. Now, we think about cancer being bad, but continue doing their unprogrammed cellular replication. And they're typically linked up to your lymphatic system, which is essentially the waste products of your immune systems here. So this is what a benign tumor looks like. Now, when we're thinking about a malignant tumor looks like, what a malignant tumor has done is essentially hopped onto your circulatory system or hopped onto your lymphatic system and being transported to somewhere else in the body that has then gone and formed a tumor somewhere else. So that's what we're sort of looking at the cellular level in terms of tumors. So with the sad part over, let's talk about the birds and the bees. Let's talk about inheritance. So we talked about, meiosis and how we get all these new things. And this all really ties to inheritance. How do we pass genetic information on from one generation to the next? When we talked about Darwinian evolution, we were thinking about this idea, right? Traits have to be passable to the next generation. And Darwin didn't have a really great way to explain what was happening, but I think you all should know by now that the mechanism of inheritance is your DNA. Your DNA allows you to put things from generation to generation to generation. And inheritance is a weird and wild thing. I mentioned that my sister and I look nothing alike, but you can look at this group of children here and they look super duper similar. And inheritance is a weird thing. It is weird. So let's talk about it now. And so remember, the idea being is that we start off with maternal chromosomes and paternal chromosomes. Gametes have half the amount of DNA from mom and dad, and those gametes fuse during fertilization to give you a child that has half the chromosomes from mom and half the chromosomes from dad. So that's just sort of the flow. Parents make sperm, egg, which makes kid. And inheritance is sort of a mixed bag. It can be complex and it can be very simple. And so in terms of simple things, there's a number of traits out there that are single gene controlled. These include things like cleft chins, how your earlobes are attached, or having a widow's peak. So these are traits that are controlled by single genes. And when we think about sort of complex things like eye color, we're looking at potentially things that are controlled by multiple genes. And so genes, again, they make the proteins in your body, but again, proteins are the action, and they ultimately are the traits within an organism. And so how these genes are inherited from one generation to the next can be very, very simple, as we're seeing here, but it can also be much more complex. And so when we're sort of looking at the underlying genetics of a population, we're looking at what's called the genotype. So whatever your type, whatever your DNA sequences of your genes are, or whatever alleles you have, that is your genotype. That's in contrast to your phenotype, which is simply how you look. And so we can think about this population. The phenotype here is white and brown, and then there's some sort of underlying genetic code here that says, okay, here's the DNA sequence that makes me brown, and here's the DNA sequence that makes me white. And this all started, this concept of heredity, this genotypes, phenotypes, all started with Gregor Mendel, and he was an Austrian botanist, and he's typically considered the father of modern genetics. Here's a nice sort of YouTube video about Gregor Mendel's life. But Gregor Mendel, like most scientists, did not work with a particularly very exciting organism to understand genetics, to understand heredity. Mendel worked with pea plants. And the way Mendel worked, the reason Mendel worked with pea plants is they were sort of ideal. Sort of the reason unspectacular organisms are good for science is because they have really good properties about them. And so one of the things that was nice about pea plants is they are easy to maintain, they're easy to breed, and they produce offspring very, very fast. So you could do experiments in a few weeks as opposed to a human population where you'd have to wait hundreds of years, and no scientist could study hundreds of years in their own lifetime, right? People just don't live that long. So something like peas allows you to study heredity, allows you to study genetics. And now Mendel, when he was looking at his peas, he's looking at traits, and the traits that Mendel was looking at were flower color, the shape of the peas that they were making, as well as the color of the peas that these organisms were making. And what he was doing is he would breed his peas till we get pure breeding populations. So the idea, he would have flowers that would, when you produce offspring, they would only make one type of offspring. So in this case, we have a pure breeding population of purple flowers or a pure breeding population of white flowers. And the only offspring they would have would be one color. They wouldn't have any mix match, meaning they only had alleles for purple color, or in this case, alleles for white color. And Mendel sort of established this concept of dominance and recessiveness. And so when we're thinking about traits in a natural population, some were more likely to appear than others. And so in the case of Mendel, what he did is he crossed his true breeding purple flower plant with his true breeding white flower plants. And what he got in the next generation, what he would call his F1 generation, but you don't need to remember that. In his first generation, he got all his offspring were purple. And that tells you, well, maybe something's stronger about the purple than the white. And so Mendel said, okay, what happens if I cross two of these first generation purples together? And what he found is that in that second generation, he had majority of these were purple, but he did have a few white ones. So what this is telling us is that there's some underlying mechanism here that's saying that purple is dominant to white. And this is what Mendel really put forth, this idea that there's some genes just essentially override other genes. So if I have this purple flower gene, it would override this white flower gene. For the same gene. So I have a gene that controls flower color, like Mendel did. A heterozygous individual has a purple allele and a white allele. This is in contrast to a homozygous, which only has essentially alleles from one type. So in this case, it's homozygous for purple. In this case, it's homozygous for white. And now the other big thing Mendel put forth is this idea of segregation. And so the idea being is that if we have a pea plant, it makes sperm and egg, right? It's a flowering plant, so it produces pollen, it produces flowers, right? So it produces gametes, just like we do. Now, the idea being here is Mendel's came up with this idea of segregation. The idea being is that and as we've sort of talked about thus far, is that each offspring only receives half the genetic material from mom and half the genetic material from dad, or in the case is half the genetic material from this plant and half the genetic material from this plant. One of the important things about Mendel's thing is that this happened randomly. So there was no like, I'm going to make sure you get this trait, or I'm going to make sure you get this trait just because one's dominant or recessive. It's all random. So whatever fertilization happens from these gametes here is completely random. So there's no it's sort of a random event. And so when we think about how this works for Mendel, he had two heterozygotes. So we had one allele for purple, one allele for white, and you have two of those together. So this would be this cross here. The idea is that the gametes that they can produce only are, this plant can produce a gamete that has a chromosome that has the white allele, and it can produce another one that has the purple allele, and the same thing for this plant. But when you take those and you essentially have fertilization of this gamete with this gamete, or this gamete with this gamete, or any of these four combinations that you see, we produce different types. And so the idea being is that we produce either, if we have two whites together, so in the case of fertilizing this with this, we get a homozygous recessive, so this plant would be white, as you can see here. We fuse this one with this one, right? Purple with white. We get this heterozygote, which again, you can see is purple because the purple is dominant to white. But again, it has one purple allele and one white allele. Same thing here if we fuse these two, or if we fuse these two, or these two. But if we take the two purples together and we create a homozygous dominant, meaning it has two purple alleles and the flower color is purple. So this is Mendel's idea, that this process is random. So which fertilization happens is random, and that there's a segregation here, that you only send half your genetic material to each of your things, and it happens randomly. You don't produce, more dominant or recessives. If it doesn't work that way. It's all 50-50 between your alleles. So half your chromosome, half the sperm any male produces will get one allele, and half will get another allele. And you can think about this like, how sex determination works. Like every time a male produces sperm, half the sperm have the Y chromosome and half the sperm have the X chromosome. So this is what Mendel brought forth, this idea of heredity and dominance and recessive. And sort of just remember that meiosis is 2 rounds of mitosis without any DNA replication, reducing the amount of DNA in half. And we talked about the terminology. And so that's what Mendel put. So let's talk about some sort of ways we apply this. And so we're talking about this idea of complex versus sort of simple traits, and blood type is one of those very simple traits. And so your blood type is controlled by a single gene. And as you all know, you can be type A, type O, type B, or type AA. And that's all controlled by one gene. And it depends on how many alleles you have. So in the case of Mendel's peas, we just go back here, there was only 2 alleles, right? One allele for flower color of white and one allele for flower color of purple. Now, blood color is a little bit more in depth. Blood type, there is an allele for type A, there's an allele for type O, and there's an allele for type B. So this one has 3 alleles for one gene, as opposed to flower color, which only had 2 alleles for one gene. And when you sort of mix and match blood colors, if you have 2 A alleles, you become type A. You have an A and an O allele, you're still type A. If you have a B and a B, you're type B. A B and an O, you're type B. If you have A and B together, you're a hybrid, you're AB. And finally, if you have 2 O alleles, you are type O. What you're noticing here is that O here is always recessive to A and B. And B and A are actually expressed together. So they're what we call codominant, meaning neither is dominant over the other. And we see the resulting phenotype, again, the phenotype being blood type, expressed essentially together. Which again is in contrast to an A and an O allele, which produces only A blood. So that's the sort of simple example of blood type. Blood type, as I mentioned, is not just A and O. I'm sure you guys all know A positive, and there's all sorts of other stuff here. If you want to sort of dive deep into it, this is a really nice YouTube video about explaining how the positive negatives work as well, but and sort of the underlying mechanisms behind this that you can take a peek at. Excuse me. But it's sort of beyond the scope here, but I just wanted to point out blood type is simple in this A, B, and O scenario here, but it can be much more complex. And so if you want to learn a little bit more about that, good YouTube video there. So next up with codominance is what we call incomplete dominance. And this is where one allele actually isn't fully dominant over the other. So the idea being is we can have a red flower and a white flower mixed together, and the allele that causes redness, which is characterized by a big R here, and the allele that characterizes white, which is a little r, is essentially one is not dominant over the other. essentially the idea being is this one only can partially suppress the effects of this one in the offspring. So the idea being is you have an intermediate where you have a mixing of these colors together. So I'm sure, as you all can probably figure out, mixing red and white gives you purple. And that's what's happening here. So incomplete dominance rely essentially produces a mixing of colors together. And this is pretty common in the flower world in terms of mixing of colors as well. As we mentioned, codominance of traits is when both are expressed together. We see this with blood type, but we also see it in colors of fur. So if you ever see a brown and white cow or a red and white cow, it has actually a codominant trait where it has alleles for both red and white, and they're expressed not together, but they're actually expressed differently in different spots. This is why you get the spotted color on cows. In case you didn't know how spotted cows are made. In addition, there's another sort of more real-world application of codominance, which is actually sickle cell disease. And so sickle cell disease or sickle cell anemia is a blood disease. What it does is it causes your blood cells to go from being their normal pancake-shaped to being this shaped, either essentially long and very thin, or sickle-shaped, like we're seeing here in the picture. And one of the sort of the side effects of having your blood cells, instead of being chunky and round and being flat or hooked like this, is that they have less capacity to carry oxygen. So those that suffer from sickle cell disease or sickle cell anemia essentially have lessened oxygen carrying capacity. They are less able to do physical activity and stuff like that. And so this is a type of codominance disease where we have the homozygote dominant here, where we have the person shows no effects of the disease. It's controlled, again, this is all controlled by one gene. We have the homozygous recessive, where the individual has 2 copies of the bad gene, which means they have the full-on sickle cell disease, meaning they have no circular blood cells circulating. They all have these sickled ones. So these people that suffer this are really, really struggling with lack of oxygen. And then finally, we have the heterozygote, which has, again, 1 dominant allele and 1 recessive allele, which means it has, and it expresses both because it's a case of codominance, meaning it has both sickled and fully formed normal. And so this is what we typically consider sickle cell trait, meaning that these people have, relative to sort of healthy individuals, have a lessened capacity to carry oxygen, but relative to the sort of people that have the full-on sickle cell disease, have a much, much greater oxygen carrying capacity. So this is sort of a real-world example of codominance. That being said, the sort of the prevalence of this disease is typically for most people living in African nations. You typically don't see it very much outside of those with direct descendantship from Africa in some shape or form. Next up is our sex-linked traits, and these are traits that are found only on your sex chromosomes. So as I mentioned, sex determination is done by these sex chromosomes, but there are a number of traits out there that are in genes that are carried on your sexual chromosomes. And so colorblind is a great example of this, where we have our X and Y chromosome, and we have a gene on there that controls color blindness. And so we have the dominant allele, which we'll characterize by big R here, produces the fully functioning proteins, meaning you can pick up all the different types of light. And then you have the recessive allele, which we define by little r here, which produces the proteins in your eyes that don't allow you to pick up all forms of light. And now when we're thinking about being colorblind, males and females behave differently. And so for a male to be colorblind, it only needs the recessive allele on its X chromosome, because the Y chromosome is actually missing that gene. Sort of one of the functions of the Y chromosome, it just doesn't carry a lot of genetic material. It doesn't carry a lot of the traits that the Y chromosome does. So if you're a male and you have colorblind, you have an X chromosome that has that recessive allele. And because you have no corresponding allele on your Y chromosome, you will thus be colorblind. That's in contrast to females, where you need 2 copies of the allele to be physically colorblind, which is why the rates of colorblind in males versus females is skewed to being heavily dominated by men being colorblind. That being said, there are plenty of cases of women being colorblind, but again, it's a trait that is mostly commonly found on men. Thinking about normal vision, males have to have a dominant allele on their X chromosome, and females can have either 2 dominant alleles or 1 dominant recessive. It doesn't really matter. This is a case of full, complete dominance. So you need two of the alleles or 1 allele plus a Y chromosome to display this trait. But as I mentioned, genetics is typically very much more complicated than one trait. So for instance, eye color is controlled by multiple genes. And so we can look at a sort of a simple cross here between a mother and a father, both with brown eyes. They're both heterozygotes, and you'll notice they produce a sort of a flurry of different eye colors, mostly browns, some intermediates, some greens, and some blues. And so this is a case where a single trait is controlled by multiple genes. Next up is actually pleiotrophy, and this will actually talk about sickle cell anemia again. And so for those people that suffer from sickle cell, whether it's the trait, meaning they're heterozygous, or the full-on sickle cell disease, they actually have this case of pleiotrophy. And so their genes that control the shape of their red blood cells actually has multiple effects. So one of the interesting things that happens is that not only does people with sickle cell have disruption of oxygen transport due to the sort of non-conventional shape of the red blood cells, they actually have decreased and almost sometimes zero capacity to carry parasites. And this is a particularly important thing in Africa, where a parasite, as I'm sure you all know, is malaria, is extremely rampant. Those that have sickle cell trait or sickle cell disease, they actually are almost physically immune to malaria, because malaria physically actually can't infect the red blood cells. So again, we have one gene controlling malaria infection rate as well as the shape of your red blood cells. So this is what we call pleiotrophy: one gene, multiple effects. In addition, environment also matters, and so your genes can be activated or turned on depending on temperatures. So cats and rabbits, depending on where they live, they can take incredibly different colors. So Siamese cats will typically get darker in colder environments and lighter in warmer environments, and we see the same thing for hares and some rabbits as well. So it just depends on the environment in this case. And so, well, actually, we're not wrapping this up. Let's keep on moving. Let's talk a little bit more about genetic disorders. And so, as we've mentioned, we have both autosomes, which is our sort of our 22 pairs, and then we have our sex chromosome, which are the sex-determining chromosomes. And genetic disorders can be either found on these autosomal chromosomes or on these sex chromosomes. Give me one moment. I have another dog issue I have to take care of. Okay, sorry about that. I adopted a new puppy this past weekend, and he requires a lot of attention and requires a lot of cleaning, including cleaning up vomit, which is gross because he eats his own poop. Anyways, back to genetic disorders. So we're going to talk about the not so fun part about genetics. I'm sure you guys have all heard of genetic disorders in some capacity, whether it's just, through media, or maybe you know someone with genetic disorder. But genetic disorders can be either carried on your autosomes, which again are the chromosomes that aren't your two sex chromosomes. They can be on your sex chromosomes, or they can be the result of chromosomal abnormalities, which we talked about with Down syndrome and Klinefelter syndrome and all the other sort of types of syndromes. And so autosomal disorders are genetic diseases found only on your non-sex chromosomes. The vast majority of sort of autosomal diseases out there are typically recessive. And so you could be a carrier for any number of autosomal genetic disorders, but not show any symptoms. And there are some out there that are dominant. We're going to talk about cases of both. So the first one we're going to talk about is cystic fibrosis. It's actually one of the most common genetic disorder among Caucasians. So about 1 in every 2,500 people suffers from cystic fibrosis. And in addition, about 1 in every 20 Caucasians is a carrier of the gene. So as I mentioned, cystic fibrosis, well, actually I haven't mentioned this, but cystic fibrosis is a autosomal recessive gene, trait, disease, words. And so you could be a mother and a father and both be carriers for this gene, but not show any symptoms of the gene, because again, it's recessive. And you could produce 4 offspring. 1 is completely normal, no trace of cystic fibrosis, 2 kids that are carriers, and 1 kid that has cystic fibrosis. You had no symptoms of the disease, but because it's a recessive gene, and because the kid can inherit a recessive gene from mom and a recessive gene from dad, it can then have cystic fibrosis. And cystic fibrosis as a disorder arises from a defect, a mutation on your 7th chromosome. It actually changes very, very minimally a gene that actually pumps sodium and chloride ions in and out of your cell. And what ends up happening is cystic fibrosis essentially outside your cell. So we have just a normal lung cell here. This is typically where cystic fibrosis attacks. It's typically a lung disease. You have inside and outside, and then you have your plasma membrane here. You have a normal protein, and then you have a mutated protein. Normal protein moves things in and out, no big deal. Now, our people with cystic fibrosis have a mutated channel, which causes mucus to build up. And what this does is it makes breathing very, very difficult for people. In addition, it can cause damage to your pancreas, which can lead to malnutrition. And in addition, people that have lots of mucus on their lungs are much more susceptible to bacterial and viral infections. So overall, people with cystic fibrosis have all sorts of issues, but it can also be much more susceptible. In particular, the people that get cystic fibrosis are really, really commonly infected with Pseudomonas aeruginosa, really, really nasty bacteria out there. But overall, cystic fibrosis typically tends to be fatal on average, your 20 to the 25th birthday. Some people can live longer, but it's a genetic disorder, which means there's no real strong medications for it. It's pretty common, but it's still something that's very hard to treat. And so it is, again, typically fatal. People that have cystic fibrosis, again, don't live very long, but those that do typically get what are called back pats to try to break up that mucus, because most people that end up dying from cystic fibrosis typically do it from some sort of lung infection. But if you can break up that mucus layer that's forming in your lungs, you can potentially prevent an infection. In addition, people with cystic fibrosis typically are on antibiotics for an extended period of time to try to prevent this disease from prevent this disease from giving them infections. And then finally, you can also take enzymes to help sort of mimic the function of your pancreas to help essentially prevent you from getting malnutrition. But again, cystic fibrosis is kind of a sad disease. You basically, either you choke to death on your mucus or you get a bacterial infection and you die. So it's sort of a really sad disease. But again, it is autosomal recessive. Moving on to an equally sad disease, and I think this one might be the saddest of all the diseases we're going to talk about today, and this is Huntington's disease. It's also formerly called Huntington's chorea. And this is an autosomal dominant disorder, in which means you only need one bad allele to get the disorder. And Huntington's is a really awful disease. It's also just a jerk of a disease, and it'll become apparent in a second why it's a jerk. But it does cause death of your brain matter, and it has very late onset in life. So typically, people that develop Huntington's and eventually die from Huntington's typically don't develop it until they're in their 30s, 40s, and sometimes even their 50s. And the idea being is that you can be a parent, you can have a partner, and you can have a child and pass that gene. If you have Huntington's disease, you can pass that gene on to the next generation. But the thing about Huntington's, which is why it's a jerk, is again, it's late onset, meaning you don't know you have Huntington's until you're in your 30s to your 40s, sometimes in your 50s. So sometimes when you're getting ready to have kids, or after you've had kids, or your kids are even adults. So you can pass this gene on without even knowing that you're having it. And again, because it's dominant, you only need one copy of the allele to do it. So if this person only has one copy of the allele, well, if one of these kids we see here gets it, he gets Huntington's. And this father or mother did not know that they had that gene, which is why Huntington's is a jerk. Because it's cystic fibrosis is, it's recessive, so you're not passing that down to the next generation. Most likely, you have it when you're born. Huntington's, you don't know it till late, and it's something that propagates itself through time because it's just sort of hard to know. Again, Huntington's is dominant, so it's typically due to heredity. But there's new research out that shows that some people that actually get Huntington's, it's this rare genetic mutation that happens on I think it's chromosome number 9 that leads to Huntington's disease forming. So it's typically hereditary, but it can be sort of spontaneous, which I think is kind of scary, personally. But those that are suffering from Huntington's again, it's a neurological disease, so they suffer pretty severe things until they die. There's no known treatment for Huntington's. There's medications out there that lessen the symptoms and prolong your life slightly, but you get Huntington's, you're not in for a good time. And there's sort of the symptoms, as you'd imagine with neurological ones, are pretty severe. So you have clumsiness, irritability, depression, severe memory loss, muscle spasms, and it really just attacks the basal part of your brain. So all your sort of normal functions go out the way. It's not like Alzheimer's where you lose your memory. You just become very irritable, very shaky. You develop what's called a chorea, which is a twitch, and it's kind of constant. So you're constantly shaking. It's sort of a really, really, really bad way to die. And not something I whatever wish on anyone. So that's Huntington's, and I said the most depressing of all diseases. Let's talk about one that's slightly depressing, but less depressing, and this is hemophilia. And so hemophilia, you might have heard of as the royal disease, and this is a sex-linked disorder. It's recessive, but it is carried on those sex chromosomes, which meaning males are going to be more likely to be affected than females. But again, it is a sex-linked. It is a clotting disorder, and it is kind of interesting. So if you want to sort of know why it's called the royal disease, take a peek at this YouTube video. It's a really sort of interesting look at how monarchies sort of pass this disease on through time because of all the so-called close breeding to keep bloodlines pure. And so, if you want to know how sex-linked, why this hemophilia is called the royal disease, take a peek at this YouTube video. But again, it's sex-linked, it's recessive. It just leads to bleeding. So those people that have hemophilia simply can't clot their blood effectively. So if you're hemophiliac and you get a bruise, you essentially bleed indefinitely because your blood just never stops. There are lots of treatments for this, by the way, so it's not really particularly fatal anymore, but you can imagine 300 years ago, it was might have been fatal. So those are diseases, but let's talk about things that are less depressing. And so let's talk about things that are interesting, things that are cool, things that, maybe have some meaning of your everyday life. And we're going to start off by talking with lactose intolerance, which is sort of an interesting thing. So you as an adult should not be able to drink milk. And as a matter of fact, 80% of the adult world physically can't drink milk. 80% of all adults on this planet have some sort of lactose intolerance. And now most people sort of don't think about it, right? You could be lactose intolerant and you could eat pizza and you might get a little tummy ache, or you might get a little itchiness in your throat. You might get something. And you might not really think, well, that's lactose intolerance. And that might never, ever not get worse, but you're still lactose intolerant. So it's not like the people that get really, really sick, right? There's sort of gradients of lactose intolerance. But the idea being is most adults on this planet are lactose intolerant. And this is all due to losing the function of one gene. So lactase is the gene that breaks down lactose into usable components. Those people that don't have or they have an inactive lactase gene can't break down the lactose or the sugar inside this delicious slice of pizza here, in particular the dairy here, and they essentially can't digest the lactose, which can lead to any number of different symptoms inside of a human body. But adults, we all know, most of us know that we still eat lactose. It's in a number of things. And the sort of interesting thing is that there are some adults, if you do your math right, that if this, you think about 80% of people can't break down lactose, well, that means 20% of people can break down lactose. So what the heck's happening here? Well, it's sort of an interesting thing. This is an evolutionary case. It's also heredity thing. And so this actually, lactose intolerance and the tolerance of lactose actually arose with the rise of cheese. And so we know that cheese, depending on who you ask and what you would consider cheese, likely became a thing in the Middle Eastern region of the world. It depends who you ask, but it's anywhere from 10,000 to 5,000 years ago. It's sort of interesting. If you start to look at the genetics of people, you start to see that with the rise of cheese, you're starting to see people eating cheese, you're starting to see mutations in the lactase genes, in particular in this Middle Eastern European area, where less people have lactose intolerance as adults than others. That's leading them to still be able to digest lactose as an adult when they really shouldn't be. Because there's really no reason for an adult human being to ever need to break down lactose. The only reason as a human you need to break down lactose is when you're breastfeeding. And that's pretty much it. So the idea being is that we had this artificial selection, this cheese, the invention of cheese and the dairy industry as a whole, that is putting a pressure on the human population to make us want to essentially have a mutation and tolerance of mutations in the genes that control the breakdown of lactose. Again, all because of cheese. So I think that's a fun one. Next one is actually our Dutch. So people that are from the Netherlands are actually typically much taller than most of the other places in the world. So the average male in the Netherlands is about 6 feet tall, which when you compare that to the global average, or even the average here in the United States, which is still about 5'7", is 8 to 5 inches taller than the rest of the world. And one of the sort of interesting things, if you look at the sort of the demographics, is that men in the Netherlands are getting taller and taller and taller. Females in the Netherlands are getting taller and taller and taller. And we've been increasing 1 inch, greater than 1 inch per decade in the Netherlands, which is really fast. So the population is getting an inch taller every decade. And then conversely, if you just think about sort of history-wise, right, people in, back in, say, 0 BC, right, were much, much shorter than they are now. Which I think is interesting. And just to put that in perspective, if you contrast the sort of how much males in the United States have grown versus males in the Netherlands have grown, males in the United States have only grown by about 2.4 inches, whereas those in the Netherlands have grown upwards of 6 inches in the same time period. And so the question we can ask is why? Well, heredity tells us that the people that are most likely to be successful in terms of producing offspring are going to pass on their genes to the next generation. And so the idea being is that being tall, and I think this is true in many cultures around the world, that being tall is a desirable trait. So women are selecting men that are taller, which means those men are more likely to pass on their genes to the next generation, which means that their children are likely to be taller, which is driving the patterns we're seeing here. Because people want to date and marry and have children with people that are taller, well, we're going to have taller offspring over time. We see this in the Netherlands. We actually see this as in a global way. People are selecting for taller individuals, not because it's beneficial, because you can, say, get fruit from a tree, but just simply because it's more attractive. And again, evolution is all about passing your genes to the next generation, shifting the distribution. Molars, is that the percentage of people with wisdom teeth is actually, sorry, the percentage of people being born without wisdom teeth is actually increasing. So previously in the 20th century, it was, 10 to 15 to 20% of people being born had no wisdom teeth. But now it's about 35% of the population actually has no wisdom teeth. And sort of the idea is that our ancestors, so if we think back to sort of when we talked about human evolution, our ancestor had a larger jaw because they had a tougher diet. And so the evolutionary answer to that tougher diet was an extra set of molars on the top and bottom, which allow you to break up that plant material, break up that flesh, right? But if you think about sort of modern humans, we don't really have that problem, right? We cook our food, and cooking your food is a great way to break it down without having to macerate it with teeth. And so there's no longer an evolutionary advantage to carrying the genes that make you have wisdom teeth, because we don't have the skull space, because most people have to get their wisdom teeth removed, but also because it's just not beneficial. So it's just a trait that's actually been lost over time. So it's not really something that was selected for in terms of reproductive success. It just wasn't a necessary trait. And as we sort of remove them and they cause problems, it became deleterious to keep them around. So we're seeing a population lose wisdom teeth simply because we don't need them anymore. Another sort of interesting aspect of human heredity and human evolution is actually what's happening to our brain size. And sort of one of the things, as I mentioned, is that humans are pretty smart, very, very smart. That's pretty much the only thing that makes us capable of being an apex species on this planet. Otherwise, we're not physically fit enough or have any great traits about us. The only thing that actually makes us good is our brain. And one of the sort of interesting thing is we look at how brain size has sort of gone through time. We look at Homo habilis, Homo erectus, Neanderthals, or Homo sapiens, including modern Homo sapiens. We see that brain size has increased and then is slightly decreased. And so sort of interesting phenomena, right? We associate with having a big brain relative to our body being a prerequisite for being very, very smart. But what we're seeing here is our human brain is shrinking. And it's sort of an interesting thing. So some people, and this is not a settled question by any stretch of the imagination, but some people say we're getting dumber. And, I can understand that from a certain perspective. But others say it's simply becoming more efficient. You don't need to have this big honking brain if you can wire it more efficiently. So think about cell phones, right? When the very first iPhone came out, it was big and clunky, right? Really thick. It wasn't like big screen size, but it was clunky, right? It was thick and it was made out of hard materials. But as the generations went with the iPhone, as they rewired it, as they improved the technology, it got smaller and smaller and smaller. Now it's, probably a third of as thick as it used to be when it first came out. And that's the idea of the human brain. You can rewire it, make it smaller, but be just as good as a larger brain. So this is a question that still hasn't been answered, but it's sort of an interesting thing. We're seeing a shrinking of the human brain overall. And then finally is blue eyes. And so eye color, as I mentioned, is controlled by multiple genes. And sort of interesting thing, if you look at sort of all the combinations of how individuals with eye color make offspring, we can look at parent 1 and parent 2 here. So you have two blue-eye individuals. They can pretty much only make blue-eyed and some green-eyed, but you'll notice they can never make a brown eye. And anytime you have a blue-eyed person present without a brown eye, they can never make a brown-eyed children. Now that's in contrast to brown and brown, which makes sometimes blue, sometimes green, and majority of time purple, brown. And so it's sort of an interesting thing, right? A blue-eyed individual can never make a brown. So what's going on here? So eye color is interesting. Eye color is just sort of this weird thing. So blue eye color is actually a mutation. It's recessive. The idea being is that when you have blue eyes, it's a lack of pigment. So there are two genes that control eye color. If you have two functioning copies, and actually the one thing I'd like to mention is if you have they're in succession. So you express this gene, and then you express this gene, and then you make the pigment. So you need both to occur in succession to make the pigment. Now, if you have fully functioning of both, you can make a brown pigment. But if you have blue-eyed, if you have one defective, you can't make any pigment because it's a pathway. And so that's why if you have two blue-eye individuals, you actually physically can't make a brown-eye individual because you have a defective pathway, because you need both of these to function to make a brown. So if we go back to this diagram here, we can sometimes make my colors are off today. You can sometimes make green, but you can never make brown simply because you have to fully complete the pathway to make brown. Whereas blue is, again, the absence of pigment. So with that, I have a nice little wrap-up video about heredity, but please let me know if you have any questions about any of this stuff, and I hope you guys all have a great week. I will see you all in class starting on Wednesday. Take care.